Hello, honors biology students. Welcome to unit number 2, cell structure and function. You can find this chapter in your book. It is chapter 4. For some of you, you may need an alternative source, another way of hearing about it, or reading about it also. So you can look and refer to chapter 4 in your textbook if you'd like. You do not need to do that if you do not want to, but it is available to you. The test will be completely from these notes that we're about to go over. So let's begin the notes for today. This section begins with cell theory, basically. A couple gentlemen, Theodor Schwann and Rudolph over here, they came up with some ideas that became what's known as a cell theory. All organisms are made of one or more cells. This is a fact of for what we understand life to be defined as. Life requires organisms to be made of cells. So all organisms, and organisms are living things, so all organisms are made of cells. Why did they call these things cells? Well, because when they were looking at them under the microscope, I believe they were looking at plant cells under a microscope, and under a microscope, I believe they were looking at cork possibly. Under a microscope, it looked like cells, jail cells. So that is the name they came up with when they first noticed them. Number 2 here, this is, in some books, this is the definition of a cell. Well, cells are the basic units of structure and function for living things, organisms. And finally, the last, number 3. Number 3 is where we may find some problems. Since all cells come from pre-existing cells, the problem arises in where did cell number 1 come from? How did cell number 1 arise? We do not have an answer to that. There is no theory that defines how cell number 1 showed up, basically. There's some ideas, there's some hypotheses, and we'll talk about those later when we talk about evolution, but those ideas have not been validated because we cannot prove how cell number 1, or even support it with data, how the first cell showed up. So that is part of the problem with the cell theory is, where did the first cell come from if all cells come from pre-existing cells? Now, size of a cell is important. Cells that are large. So let's look at this. This is a setup here showing you one large cell. Okay, that's 30 by 30 by 30 micrometers. That's what that symbol means, micrometers. So it's a 30 by 30 by 30. Then they're showing you another example of a cell which is made up, in this case, 27 separate cubes combined become a 10, 10, 10, 10, so a 30 by 30 by 30. And what you need to know for a cell to function efficiently, it has to have sufficient surface area, okay, in comparison to its volume. All right, it sounds kind of confusing. I'm going to show you a math question and a math to show you how this works in a second. So here's what you really need to know. The surface of a cell is the plasma membrane. It's the outside of a cell. The structure, the plasma membrane, allows things to enter and leave the cell. It controls the passage of materials. Basically, cells require plasma membranes to do that for them. If they do not have sufficient plasma membrane, they cannot feed themselves or excrete waste at an efficient pace, and that could lead to toxicity or potential death because nutrients aren't getting to where they need to go. All right, so cells that have what's called a large surface area to volume ratio are considered smaller, and I'm going to show you an example in a second, and they quickly and effectively, that's the key, they're efficient at exchanging materials, meaning moving materials in and out, and distributing, moving things around within a cell. So if this was one giant cell and it needed to move something from one corner to the other corner, it potentially could take too long. Or if something was right in the center and it needed to leave because it was a waste product, it could take potentially too long to leave, creating toxicity within the cell. Small cells don't have that issue. Things can leave and enter pretty quick, but it all comes down to something that's very important. It has to do with not just surface area, how much surface area there is in relation to the volume. And I'm going to show you right now some math problems, some math to show you how it works out. So here we go. This says, what happens to surface area to volume ratio as a cell gets smaller? So here's a large cell. So 3 by 3 by 3, all right? The surface area, calculate it out. There's 6 sides to a cube. So an area of a side is length times width. So 3 times 3 is 9, times 6 is 54. So the total surface area is 54 units, okay? This one here, this is a 2 by 2 by 2 square. On this one, the surface area, the total surface area, 6 surfaces times area of a side times 2, that's 4 times 6 gives us 24, okay? The smaller cell, a 1 by 1 by 1. So 1 times 1 is surface area of one side. 1 times 6, 6 sides. So the answer is 6. So the smallest cube has a 6 surface area. The medium one is a 24. And the large one here has a 54. Okay. Then we calculate volume. Length times width times height. 3 times 3 times 3, 27. 2 times 2 times 2, 8. 1 times 1 times 1 is 1. Okay, so what determines how efficient a cell works has to do with a ratio. You take the surface area, you divide it by volume, and you get a number. The larger the number, it tells you that the cell is more efficient. And I'm going to explain to you what that means. So for example, this one here, 54 divided by 27 gives you 2. So this means it has twice as much surface area as it does volume, so to speak. All right. This one here, as it gets a little smaller, this one has 3 times the surface area to accommodate the volume. Okay. So that means there's more surface area to accommodate volume. This last one, the smallest one, 6 times the surface area to accommodate volume. So what you need to know, and you're going to learn this in chemistry next year, if you want to increase the rate of something, you need to, one of the ways of increasing the rate of something is increasing or exposing more surface area to it. Well, as a cell gets big, its surface area does get bigger. It goes from 6 to 24 to 54. Surface area does get bigger. Okay, the surface area does not grow as quickly as volume does as it gets bigger. Okay, so if you notice 6 to 54, that's 9 times. 9 times 6 gives you 54. 1 to 27 is 27 times bigger. So the volume increases at a much more rapid rate than the surface does. So in order for a cell to be efficient, it has to have sufficient surface area to accommodate its volume. And that is why we do a surface area to volume ratio. The larger the number that you get, that means that cell is probably going to be the smallest, and that also means that that cell will also be the most efficient. 2 types of cells that you need to know about: prokaryotes and eukaryotes. Sometimes we say prokaryotic cell instead of prokaryote. Sometimes we say eukaryotic cell instead of eukaryote. Same thing. If you're talking about prokaryotes or prokaryotic cells, same thing here. What distinguishes them? They do not have a nucleus. Okay, and they do not have what's called membrane-bound organelles. So around every cell, every living thing, every cell has a membrane around the outside of it that controls what enters or leaves the cell. Well, bacteria or prokaryotes, they don't have additional membranes on the inside of them. Okay, so they don't have any parts on the inside of them that have additional membranes. So that means they don't have membrane-bound organelles or cell structures on the inside of them. But they do have their membrane on the very outside that protects them and allows and controls the entry and passage of materials. Okay, what do prokaryotes contain? Well, they don't have a nucleus. They have this thing called a nucleoid. I'll show you a picture, a different picture in a second. Which organisms are prokaryotes? Only 2 kinds: archaea and bacteria. And they're both, I call archaea bacteria sometimes, but they're primitive bacteria, so to speak. All right. What is unique about them? Well, they have ribosomes, and so do eukaryotes. They have ribosomes also, but they're a little smaller. Okay. Bacteria are susceptible to antibiotics. Antibiotics only kill bacteria. Okay. They will not kill your cells. They will not kill viruses. They only kill bacteria. And prokaryotes, they usually have a cell wall, which is a structure around the outside of the membrane. I'm going to show you a picture in a second. And prokaryotes can have a flagella. That's its tail here. These little things here, they look like little hair-like structures. They look like what's called cilia. They are not cilia. They're called pili. These are ways for bacteria to communicate and take DNA and information from other bacteria around them. So these are not cilia. They do have flagella. They allow them to be mobile and move around and swim. All right. Over here, I'm showing you bacteria that are green. There's a group of bacteria that are green. They're called cyanobacteria. They're special prokaryotes that can do photosynthesis. They have chlorophyll, so they can do photosynthesis. Now, here's a picture. We've drawn it at Valhalla to make it a little simpler so it's easier to see the structures. The space, the nucleoid region, bunch of DNA kind of twisted around there. A cell wall's on the very outside. Okay. Ribosomes are these little dots. They're protein factories, and the plasma membrane sits inside of the cell wall. And here you have a flagella, a little tail. They also could have those little hairs called pili, but I'm not going to ask you that on your test. And here's a picture without the labels. Eukaryotes are eukaryotic cells. Well, if you're not a prokaryote, you got to be a eukaryote. That's pretty much all that you have left to be. So eukaryotic cells are cells that are a little bit more complex. They have membrane-bound compartments inside of them. They have a nucleus inside of them. And what is unique about the nucleus has its own membrane around it. So it is a membrane-bound organelle. Okay. Over here, these are membrane-bound. This is membrane-bound. This is membrane-bound. Let's see what else we got here. These are membrane bound. Lysosomes are membrane bound. So you have a lot of membrane bound parts. If it has a membrane around it, you will not find it inside of a prokaryote or bacteria. You can only find it inside of a eukaryote. Here's a picture showing you the parts and what they're called and what they do. All right. So I'm going to go on the next page, which is a little cleaner to look at. We'll go here. Whoops. We'll go here to the next page, showing you what they all are, okay? So this is them here unlabeled. Here you have them labeled. The outside is a plasma membrane. Over here, the outside is the cell wall. The second line in is the plasma membrane for the plant cell. Smooth ER, is that what that stands for? Rough ER. They both have rough ER, which are these little dots attached to these things. Explain to you what they do in a second. These are the transportation tunnels or networks of the cells. Some of them look rough in texture. That's why it's an RER. Some of them look smooth. That's why it's an SER. And the rough is these little dots attached to it. The cytoplasm is the space inside there. The mitochondria are in both cells, or these little powerhouses of the cell, the energy makers of the cell. They are membrane bound, so you're not going to find them in a bacteria. Membrane bound Golgi, you're not going to find in a bacteria. RER and SER, membrane bound, you're not going to find them in bacteria. You will find cytoplasm in a bacteria or prokaryote. Nucleus, you're not going to find in a bacteria, you're going to find a nucleoid. A nucleus is a membrane-bound compartment, basically. Inside of it is a bunch of DNA, that's this twisty stuff, and you have these really dark areas called nucleolus. This nucleolus has a unique job, and we'll discuss that in a second. All right. Centrioles, these are involved in cell division. They're only found in animal cells. Plant cells don't have them, and they still don't have any problems dividing. It's interesting, but centrioles are not necessarily needed. They just help and aid in the act of dividing. Lysosomes down here, I'm going to skip the lysosomes because these are also found only in animal cells, not in plant cells. Okay. The nuclear membrane, this is the membrane that surrounds the nucleus, and there's little pores in it, little holes that allow things to leave and enter. All right. Two parts that are found in a plant, the chloroplast and the cell wall. Those are found in plants and algae. Algae are the green stuff you see floating around the ocean that do photosynthesis. They're similar to plants, but they're not in the plant kingdom. They're in the protist kingdom. Lysosomes, once again, already discussed them, only found in the animal cell. Ribosomes are these little dots. Sometimes they're free ribosomes and not attached to anything. Sometimes they're attached to structures, and those are ribosomes that are attached to the RER, okay? And finally, the vacuoles are little storage tanks. Plants have a large central vacuole, much larger than the animal cell. All right, pictures of them unlabeled. All right. Cellular organization. So we had something similar in our first unit. Our first unit was down here talking about atoms, molecules, and cells. So now we're going to stretch it a little farther. From cells, you put cells together and you make tissue. All right, a bunch of tissue that has a function becomes an organ, and an organ, a bunch of organs create a system. This is the digestive system. It involves the large intestine, the small intestine, the stomach, the liver. You also have the pancreas, which you can't see, which is back here somewhere, and those are all part of the system. And organ systems combined make up the organism. Right? The smallest living unit of life is a cell. Okay. Let's discuss a few of the organelles, and then we'll stop there, and then we'll talk about a few of them today, and then the second part will continue on. All right. So let's begin with the plasma membrane. So this is important. The plasma membrane controls what enters or leaves the cell. Here, I'm going to show you a really fancy picture of it. Look, this is a fancy picture of the plasma membrane, a three-dimensional view of it. It creates a wall, basically, for your cell. All right. Let's go back here. So what is it? It's a barrier. It creates a barrier that is hydrophobic, basically, preventing things that are hydrophilic or polar from entering or leaving on their own. All right. So since most things in nature are going to be hydrophilic, you have a lot of things that could enter or leave the cell. So you need to keep things where they belong. So membranes, they kind of create like bags or space areas where you can store things. So a cell is a bunch of stored stuff inside of a membrane or a bag. These membranes are bilayers. There's one area, a second area. Over here, one part of the bilayer, down here, the second part of the bilayer. This right here is one phospholipid. There's another phospholipid. They come in pairs. The wall that prevents hydrophilic things from entering and leaving is right here. Okay, this is the lipid. It's the wall. Prevents things from passing through this wall. So this shows you an example of a membrane, basically. You can have stuff inside there, and as long as the stuff inside here is hydrophilic, it will not be able to leave this bag or this membrane. Something hydrophilic will not be able to enter it. The very edge of it, the phosphate area, the phospho part of it, is hydrophilic. It's a hydrophilic area, and the inside area is hydrophobic. Let me move this other way so you can see it. So this internal area, these tails are hydrophobic. They create the wall. Okay? The hydrophilic head allows it to immerse itself in this watery environment externally and internally. Now, membranes, they are selective or semi-permeable. Well, what does that mean? That means that only certain things can pass through the membrane. Okay. Well, what can pass through the membrane? Well, things that are non-polar, hydrophobic, they can go right through the wall. They can pass right through. This is how oxygen enters our cells. It enters our cells directly, right through our membranes of our cells that need oxygen. When we excrete carbon dioxide, this is how carbon dioxide leaves the cell. It can excrete, it can go right through the wall. It doesn't need any special helpers to get through the wall. How can things enter a membrane that are polar, for example, the opposite, or hydrophilic? Well, hydrophilic things, they can't go through the wall. They have to go through proteins. So there's special proteins that serve as channels or transport proteins that allow things to enter and leave the cell. So what can cross a plasma membrane or the wall of a membrane? Things that are hydrophobic and nonpolar can go right through those nonpolar tails. Okay. Things that are polar cannot pass through. They get stuck. So they need a protein to facilitate their transport, basically. Okay. These membranes, they're very liquid-like. So they call it a fluid mosaic model. It's almost like it's flowing. Sometimes some cells don't want to flow too much. It becomes extremely liquid. Certain plants and even us, what do? Well, we produce cholesterol. It's lipids. It kind of thickens it up, makes it not so liquidy, not so watery-like, and more oil-like, so it doesn't flow so easily. Okay. This shows you a picture of a membrane. It has all these blue things here are proteins. Some are attached to the periphery. Some go right through. Some here are embedded inside of it. Some make a curly Q as they go through. This one is producing, this is a transport protein over here showing you a passageway. It's a type of, it's a transport protein. This one's specifically called a channel protein in this case. Receptor proteins, these help signal the cell. They let it know what's going on. All right. Marker proteins, they identify a cell. This is how if you have a transplant and you get a foreign cell inserted in your body, if your body rejects it, it's because your immune system does not notice the marker proteins as belonging, so your own immune system will attack it. So these tell a cell that it belongs there. In previous chapter, I told you guys that the cell membrane is primarily made up of lipids and proteins. These blue things are proteins, and these are lipids. These little dots, these little circles attached to these proteins, these are carbohydrate chains. They are not a main function or a main feature of the membrane. They are usually attached to the surface. These little things here that are embedded within the membrane, this is cholesterol, so it makes it not so thick. So cholesterol is a lipid. A lipid can embed itself within other lipids. All right? So you have peripheral proteins on the outside. What do these usually do? They're usually enzymes, proteins that are facilitating chemical reactions. Okay, that's generally what they do. Integral proteins are embedded in the layer. They're embedded within the lipid part of it. So this one here, not embedded in the lipid area. These ones embedded, embedded, embedded. Those are integral proteins. This is a peripheral protein. Cytoplasm. This is everything inside of the membrane and outside of the nucleus. It does not include the organelles. It's that jelly-like, liquidy space inside of the cell that organelles are generally floating around in for the most part. All right, we're going to stop there. Whoops.